This video is going to be about how to identify PVY strains using conventional RT-PCR. Potato virus Y is the biggest concern facing seed potato certification. PVY affects potatoes in two ways. One, it can decrease yield, and two, it can cause potato tuber necrotic ring spot disease. But did you know that PVY is actually made up of many numerous strains? And that only some strains induce PTNRD in potatoes? That is why a grower might want to find out the identity of a PVY strain in their field. One of the services Idaho Crop can offer is PVY Strain ID, and I am going to walk you through the process. First, we are going to start out by doing an RNA extraction. I will be using a commercial RNA/DNA extraction kit. We are going to simply follow the protocol that comes with the kit. First, you're going to want to set up your tubes like so. We're going to start out by adding a small amount of sterile DI water to some grinding bags. For leaf tissue, I stack about 3 to 4 leaves and rip about a thumb-sized piece. Then we will put it in our grinding bag in front of the mesh. For tuber tissue, I collect about 4 to 5 cores from the eyes. Put those cores in our grinding bag in front of the mesh. Then we will use a drill press to grind the tissue. The drill press has a homogenizer attachment that has rotating ball bearings. As you can see, the tissue is easily macerated. Add 540 microliters of lysis buffer into some empty tubes. Next, add 60 microliters of the sample. Be sure to remove the sample from the back of the mesh to avoid debris. The lysis buffer will help open the plant cells, which will release the plant virus. Gently flick the tubes to mix. Place the tubes on a tube rack. Invert to mix. Now we will place the tubes in a 55°C water bath and incubate for 5 minutes. At the half mark point of the incubation, you just want to invert the tubes to gently mix. Now we are going to take our lysate solution and put it in a filter column. Next, we are going to load our tubes in a microcentrifuge and spin it for 17 Gs for 2 minutes. Once the spin is complete, remove your tubes from the microcentrifuge. The filter column is designed to separate the plant debris while the virus RNA filters through. Next, we are going to add 500 microliters of ethanol to some empty 1.5 ml tubes. Then we will take our lysate and add it to the ethanol. You may discard the filter column. Gently invert to mix. This is going to precipitate our RNA, which is going to make the RNA into a more solid substance. Now we are going to take that solution and put it in a spin column. Spin the tubes at 3.5 Gs for 1 minute. The RNA is now bound to the spin column and we can discard the flow through. Next, add 400 microliters of the wash solution to the spin columns. Spin the tubes at 17 Gs for 1 minute. Discard the flow through and repeat the washing step 2 more times. Remove the spin columns and put them into a new tube. Open the spin column lids. Set a pipette to 50 microliters and add 50 microliters of the elution solution to the spin column. Be sure to saturate the membrane at the bottom of the spin column. Close your spin column lids. Load your tubes in the microcentrifuge and spin for 0.2 Gs for 2 minutes. Then spin your tubes for 17 Gs for 1 minute. When the final spin is complete, you may discard your spin column. And the final product is your RNA extraction. Prior to running RT-PCR, we must first denature our RNA. Denaturing is when we subject the RNA to heat to produce single-stranded RNA. Start out by adding 11.75 microliters of nuclease-free water into the wells of a PCR plate. Then take 4.5 microliters of your sample and add it to the well. Here we are going to add some controls. I have 6 various PVY strains courtesy of Alex Karasiv and a healthy control. Now we are going to cover our PCR plate with plastic film. Using a rubber roller, roll the plate to ensure a tight seal. Remove the plastic flaps. Briefly spin the plate to collect the water and the sample to the bottom of the well. Open up the PCR program on the computer and select the 70C for 5-minute incubation protocol. Open the thermocycler lid and place your PCR plate in the thermocycler. Close the PCR lid and click Start. Now we are ready to do reverse transcription. First, we will make our RT master mix. The volumes will be dependent on how many samples you have. Start out by adding the RT5X buffer. Next, add the 10 millimolar DNTPs. Next, we are going to add our random hexamers and oligo-DT. Now it's time to add our enzyme, reverse transcriptase. The liquid can be quite viscous, so pipette up and down to mix. And lastly, we will add our RNase inhibitor. This solution is also viscous, so you will need to pipette up and down to mix. Gently flick the tube to mix. After your samples are done denaturing, gently remove the plastic wrap from your PCR plate. Be careful to not splash the samples inside the wells. Next, we are going to aliquot 8.75 microliters of our master mix into the wells containing the denatured DNA. You may use the same pipette tip if you do not touch the wells. Once again, we are going to cover the plate with plastic wrap and spin the plate down. Open up the PCR program on the computer and select the RT-ST3 protocol. Open the thermocycler lid. Put in your PCR plate. Close the thermocycler lid. Select Start. This step will create a complementary DNA strand using the RNA as the template. Now we are ready to proceed to PCR. This will be the master mix protocol for our PCR. Start out by adding nuclease-free water. Next, add the 10x TAC buffer. Then we are going to add the 10 millimole DNTPs. Next, we are going to add the primers. This working solution actually contains 12 different primers. The final ingredient is the green TAC DNA polymerase. This liquid could be viscous, so you need to pipette up and down to mix. Invert the tube to mix. Aliquot 17 microliters of the master mix into a new PCR plate. Gently remove the plastic film from your RT plate. Set a pipette to 3 microliters. Transfer 3 microliters of your sample from your RT plate and transfer it to the PCR plate. You may reseal your cDNA and store it in the fridge if you wish to revisit it. Cover your PCR plate and spin it down. Open up the PCR program and select the PVY Strain ID protocol. In this step, the primers will bind to specific segments of the PVY strain if it's present and multiply those segments to produce PCR products. The final step is to run your PCR product on a gel. The recipe will depend on the size of your gel box. First, set up your gel box. To make our gel, I'm going to add X amount of 1x TAE buffer to a glass flask. Then I'm going to measure out X amount of agarose. The weight should be 1.2% of the total volume. Add the agarose to your flask. Put the flask in a microwave and microwave for a few minutes until the agarose is completely melted. The flask will be hot, so be sure to use oven mitts when removing the flask. Give the flask a swirl to mix. Be very careful of the flash boil when doing so. As you can see here, the agarose has not melted. Microwave the flask for longer. Here, the agarose is completely melted and now it's ready. While the agarose is hot, we are going to add X amount of CyberSafe DNA gel stain. The amount will depend on your gel size. Swirl the flask to mix. Pour the hot mixture into your gel cast. Remove any air bubbles with a pipette tip. Cover the gel with aluminum while it solidifies. In the meantime, set up your gel box. Fill the gel box with 1x TAE buffer. Carefully remove the plastic film from your PCR plate. Set a pipette to 3.3 microliters. Now we are going to add the 6x loading dye to our PCR product. You may use the same pipette tip if you do not touch the wells. If the dye does not reach the sample, you may tap lightly to mix. Once your gel has solidified, you may remove the combs and remove the tray from the gel cast. Sometimes it may be helpful to put a contrasting color underneath the gel box so you can visualize the wells. Submerge your gel tray into the gel box. Make sure that the wells are closest to the negative electrode. You may need to add additional 1x TAE buffer to completely submerge the gel. Now you are ready to load your gel. Set a pipette to 10 microliters. Pipette up and down the well to mix. Suck up 10 microliters and gently add it to a well. After you've loaded your samples, you will need to load a ladder. The ladder helps you determine the size of your PCR product and should be loaded on every comb. Put the lid onto the gel box. Make sure that you match up the right electrodes. Connect the gel box to the power source. Turn on the power source. For our gel box size, we are going to set it at 150 volts for 30 minutes. Bubbles should be arising from the electrode. Since DNA is negatively charged, the PCR products will run across the gel towards the positive electrode. The agarose produces a porous matrix in which the smaller PCR products will move further than the larger PCR products. Once our gel is done running, turn off the power source. Open the gel box lid. Remove the gel tray. Gently put the gel on a specialized imager tray. This specialized tray is compatible with our imager model as well as the DNA stain we used. As you can see, we have some excess gel. We are going to simply cut it off and discard it. Smooth out any bubbles. Open the gel imager program on the computer and select Protocol 1. Place your gel in the imager. Start the protocol. Once the machine is done, it will take a picture of your gel and it will pop up on the screen. Different banding patterns will differentiate different strains.